Okay, so as I was saying, the scientific method has a series of steps, and here they are. Funny thing is, research into cognitive theory says that for you to be able to remember something and commit it to long-term memory, you have to see or be exposed to the information 7 times. So here's one, actually 2 so far. We are going to look at problem or question, observation and research, formulating a hypothesis, the experiment, collecting and analyzing the results, the conclusion, and the last step, without which all of this would be for nothing, would be to communicate the results, right? Because if you don't communicate, then other people are going to do the same thing and time's going to be wasted. Anyway, the very first thing that we do is we look at something, like you have a problem or a question. And in order for it to be valid through the scientific method, it has to be testable. It has to be solved through experimentation. The next thing is to make an observation and do some research on that topic, because if you don't do research, like I was saying, your a guess is just going to be a guess, your hypothesis, instead of an educated guess, which is what we call it. And what is the next step? Well, if you remember, see, you haven't seen it enough to remember this, but it is hypothesis. To formulate a hypothesis, it is to predict a possible answer to that problem or question after doing your research. Example, if the soil temperature rises, then the plant growth will increase. Notice here we have 2 things underlined. Usually, whenever we formulate a hypothesis, the wording includes both the dependent and independent variable. And we will talk more about that later. But the experiment is next, right? And in order to do that, you must develop and follow a procedure. There are a lot of things that need to be said about making an experiment, because an experiment is not just random. It needs to be very well planned, organized, and everything else. So we'll talk more about that too. You need a detailed list of materials. That way, everything will be the same. And the main thing is that whenever you do this experiment, your outcome must be measurable. That means it needs to be, you need to be able to record data about it. It has to be quantified somehow. Once you collect your results, that is, as you do your experiment, right? You're going to be collecting results. Then, once you do all of that, you can either see if it's working or not by analyzing it. And if needed, you can modify your procedure at this point. And then, of course, you confirm your results by retesting. You cannot base an answer to something that important on the results of one experiment. There is no way. And of course, you usually include tables, graphs, and/or photographs. And then once you've done all of that, you analyze, you look, you graph, then you can draw a conclusion. And usually the conclusion tells whether you supported or not your hypothesis. So, and then the other thing that you need to do is to make recommendations for changes or improvements and further areas of study that you could think of. And the last thing, of course, is to communicate your results. You need to be able to present the project to an audience and expect questions from that audience. Now, if you are not doing that as a paper, right? If you're doing it in person, then yes. But if you're doing it as a paper, it's basically the same thing. You have peer reviewers. The paper will come back to you with some changes. So that is expect questions from the audience would be the same thing, right? And once you do that, once you review it, add whatever they want in, then you're published, you're a published author. Or you have a nice little project and a presentation to do. Okay, now, can you name all 7 steps? I'm giving you wait time so you think through them. And then I'll let you see them one more time. So this is number 4. You've been exposed to these 4 times now. 1 with your own brain. Hopefully you can remember them. Okay, now here you go. A look at the 7 steps again. Okay, that is all of them. Now let's look at how we would apply them to a kid doing a science project. All right. First thing, John is watching his grandma bake bread, and he asks her, what makes it rice? The grandma explains that yeast releases a gas that as it feeds on sugar that is known as fermentation, right? And so he goes and does a question. His first question is, I wonder if the amount of sugar used will affect the size of the bread loaf. Here you go once again. This is a question, but both of them include the dependent and the independent variable, which we will talk about once here in a minute. Now, a word of caution. A lot of you used affect and effect as one thing. Affect is a verb. Effect is a noun. So if you were to formulate this hypothesis and say the effect of sugar amount on the rising of bread, it would be incorrect. You have to say the effect of sugar amounts on the rising of bread. Or you could also word it to use a verb. How does sugar affect the rising of bread? All right, so once he does that, he is doing research. He goes to look at fermentation and baking and figures out the next step, which is his hypothesis. Now, this experimental design diagram is very helpful because, as you can see, there's the independent and the dependent variable. Like I said, we will talk more about that shortly. But here we go again. We have all of these. Okay, so now he formulates a hypothesis. And it says, if more sugar is added, then the bread will rise higher. And that sounds sound, right? Sounds like a good hypothesis, and it's very good. All right, once he does that, you notice it says, it's an educated guess about the relationship between the independent and the dependent variables. So when you have a hypothesis, you have both. They're always there. Now these variables, like I said, I can define them here shortly. Now, do you know the difference? If you do, that's good. If you don't, here we go. Independent variable. It is the one that the researcher manipulates or the experimenter manipulates intentionally. It is changed intentionally by that person. Just remember that. The next. So in John's case, he's going to use 25, 50, 250, 500 grams of sugar in his experiment. That's a lot of sugar. The dependent variable is the responding variable, or basically is the one that, as the independent changes, the dependent reflects that change as a result. Okay, so in this case, that would be the size of the bread loaf, correct? All right, so let's keep going. Next thing is now he needs to figure out a procedure and a list of materials. And also something very important, though not always necessary in every experiment, in every research. I know because I've done this, but in your case, it is important to have a control group if you are in a, in a lab setting or whatever. You need a control. And in a science experiment, it's the group that serves as the standard for comparison. What does that mean? Well, it could be a no treatment, or it could be one selected by the group. Okay, I mean, by a group selected by you. Okay, by you, by knowing what things are. For example, the control group for that first question we have, if the temperature in soil changes, then plant growth will, okay. Well, if you already know that the plant, it would be dependent on the type of plant you were talking about. Because if you were talking about a tropical plant, obviously you wouldn't put the control group as 0 degrees Fahrenheit. I mean 0 degrees Celsius, because that would be way too cold and that plant would die. So you would want to do something that is normal for that plant. Since normal temperature in the tropics, it's warm, then you would want to do like a room temperature, a normal room temperature, or maybe a little warmer than room temperature, which is between 22 and I would say 30 degrees Celsius. I mean, yeah, Celsius. And that's what you would want. Okay. If it was something about the bread, you cannot say absence of yeast is what, or absence of sugar, because then the yeast wouldn't have anything to feed on, and that would be incorrect. You have to use the one that has been tested already, like his grandma's recipe, right? It's tried and true. Might as well use it. And that's what she's always used, is 50 grams. So John is going to use that amount as his control. Now, there is something very important. As you are measuring one variable, you must, and I mean this is a big must with capital letters underlined and in different color. Keep every single factor the same, because you do not know what the dependent variable is responding to if you change more than one thing. Okay, so everything has to be the same. Can you think of any constant that would be helpful for John in this experiment? Think about it. Make a list. Okay, once we thought through it, let's look at the answer. All of those things and more have to be kept the same. That's why you need a list of materials. Now, another thing is the procedure needs to include at least three, and this is what we're saying, at least three of each variable. So in other words, he's saying 50, 100, 200, like that. So each time he has to bake at least 3 loaves, because you need to be able to replicate that answer. If you are going to do that, then you must have multiple trials. And he has chosen 3, which is wise, because otherwise that's a lot of bread to be baking. So he now creates a table, carries out his experiment, logs all the results into the table, and this is what it looks like. If you pay careful attention to the average size versus the amount of sugar, the grandma's 50 is 1260. The less sugar obviously yielded less, a smaller bread. 100, still smaller. 250, smaller yet. And 500, even smaller. So something went wrong. His hypothesis was not supported by this experiment. However, he notices one thing. The first 2 worked. And they did indeed decrease instead of increase like he expected. So he realized that what he's done is he has added way too much sugar. And he now wants to retest. And he starts to think, okay, if 100 grams from 50 was too much already, then I need to lower my numbers. And so he chooses amounts between 50 and 100. And there it goes. And he carries out that experiment again. And here are the results. Now, can you tell which group did the best? Very closely, very easily, right? I mean, you can see how you got 50 for grandmas at 1344. 60, we have 1380. 70, we have 1612. 80, we have 1332. Still above grandma's, but I mean, not above grandma's, it's too much already. And 90 is 1084. So his hypothesis is supported. However, there is a point at which the size does not respond anymore. That is once he has reached what is known as the optimum amount of sugar. Anything beyond that is going to decrease. So that would be correct, 70 grams. So he finds out that that makes the largest loaf, and his hypothesis is accepted, but he also needs to now tell his grandma, and maybe she'll modify her recipe so that her breads will be even bigger, her bread loaves would be even bigger. So you see, even an experiment that is not what you expected tells you something about what you're trying to find out. So there really are no bad experiments in science, just results that don't go with what you expected. And that is it. Now, keep on observing the world and using the scientific method. All right, so that is the end of our lecture for today. We are talking about scientific method, and I am now closing off, and we are seeing you next time in person, Wednesday. Bye-bye.